Right, quick run through gene expression and epigenetics now. So, control of gene expression is our broad theme today. We're talking about gene expression in terms of when a gene is expressed. That means it's getting transcribed, it's getting translated, and will therefore form whatever proteins are coded for in that sequence of DNA bases. Okay, so genes can be turned on, they can be turned off. And one of the things that controls whether a gene is expressed or turned on is epigenetics. Okay, the epigenetic markers are this single layer of chemical tags on the DNA. So that's what the epigenome is. There's two types of tags. They're methyl groups or acetyl groups. And epigenetics is the study of these heritable changes in function, or inheritable changes of whether you'll express your gene or not, without changing the DNA base sequence. So they're chemical markers attached to the DNA or attached to the histone proteins rather than the actual DNA molecule itself. So, two different groups. If there's an increase in methylation, that means there's more methyl groups stuck to the DNA. This will inhibit transcription, so the gene won't be expressed. Why this happens is to do with the charges of the molecules. So the histone proteins I've drawn here in purple are positively charged. The methyl groups are also positively charged. Okay, the DNA is negatively charged. So by having these opposite charges, they attract each other, and by having extra positives, that means the DNA is more tightly wound around the histones. It's attracted more strongly, tightly wound around the histones, so there's nowhere for RNA polymerase to join on and transcribe that DNA sequence into messenger RNA. So it can't be transcribed onto messenger RNA, it can't be translated into proteins, and those proteins will not be made. So that gene is not expressed. The other group are acetyl groups, and if there's an increase in acetylation of the histones, that means there's more acetyl groups joined to the histone proteins this time, not the DNA itself, so the proteins that the DNA winds around. This means that transcription is more likely to happen. It allows the genes to be expressed. So again, to do with the charges, the histones are positive. We've got these negative acetyl groups stuck onto those histones. The DNA is negative, so negative and negative repel. So what we've got is these acetyl groups actually mean the DNA isn't very tightly wound. We get loose bits of DNA that can be transcribed, translated, and made into whatever proteins they code for. Okay, so just to recap that, I made a couple of little models out of the kids' toys. So here, the magnetic blocks that are purple are the histone, the green, minty green, are the methyl groups. The DNA here is getting attracted, so the steel wire is getting attracted to these methyl groups and histone proteins really tightly, so it's really tightly wound. That means I can't get my RNA polymerase to join anywhere to transcribe that gene, so it won't be expressed. The other option is that the histone proteins have these acetyl groups on them. They actually mean that the DNA isn't as attracted to the histone proteins. So by covering them in a non-magnetic material, I'm trying to demonstrate that, the DNA then winds around the histone proteins much more loosely, so it can be transcribed because messenger RNA can be made when RNA polymerase binds on. That's what my peg's representing. So it can be transcribed, would make a messenger RNA molecule that can disappear off to the ribosome and be translated into the functional protein. Okay, so depending which genes a cell is expressing or not expressing, depends which ones will make messenger RNA, which will make a protein, and therefore what that cell will be like. Okay, I hope that helps. Take care.